final day of oral presentations. Our first presenter is Penny Chan. You are welcome to come up.
Good afternoon, everyone. My name is Penny, and I'm a research officer from Bioinformatics Institute. And this afternoon, I'll be sharing more on our ongoing collaborative project on the genomic insights into COVID-19 susceptibility and severity among Singaporeans. So I would firstly like to start off with addressing the issue of heterogeneity and disease severity from COVID-19 infections. So during the COVID-19 pandemic, we have observed that a person's response to the infection can vary quite tremendously, such that some people, they are asymptomatic or have mild symptoms, while other people, they can become severely ill or even succumb to the disease. So there are many factors relating to the host, to the virus, to the environment, that could address this complexity in COVID-19 clinical phenotypes. So the differences in genetic makeup of the host is one such factor that could affect a person's response to infection, leading to the wide variance in infection outcomes. So finding the relationship between host differences and infection outcomes could help us to improve our disease surveillance and personalized medicines in the future pandemics. So our project is a multinational platform initiative, and it involves collaboration between A*STAR, SG10K Health, Prepare, and MOH Trust. So if maybe some of you may not be familiar with Prepare and Trust. So Prepare is a national program set up by the Ministry of Health, and it is for the purpose of facilitating research in epidemic preparedness. And Trust is a national data sharing platform also developed by Ministry of Health to designed to facilitate the sharing of anonymized healthcare related data for the purpose of research. So Dr. Naja and myself from BII, we have worked very closely with Professor Kelvin from Prepare and his team members, Wei Hao and Jeremy, in this project of ours, which aims to investigate the host genetic factors and identify the biological mechanisms behind the heterogeneity of COVID-19 severity, specifically in our local population. So in our project, we have three main objectives. The first one is to survey published genetic variants that are related, linked to the COVID-19 susceptibility and severity. And then secondly, we would want to run the genome-wide association study in using our local data set, the SG10K, in order to identify the novel genetic factors that are associated with COVID-19 severity and susceptibility. And thirdly is to develop the genetic risk scores, and this would enable us to evaluate the genetic predisposition in our local population and allow us to identify subgroups of individuals which are at higher risk of developing severe COVID. So from surveying the published genetic variants, we data mined and we listed over 4,000 variants across 31 publications. And these publications are reporting GWAS results, predominantly from European populations. And out of this list, we further shortlisted and focused on the variants that had the available risk allele frequencies, as we would want to follow up with a comparison of these allele frequencies against those that we have in our local data sets. So here is a map plot that shows the data mine severity variance, and it also highlights to us the loci which are containing variants that are highly reproducible, where we can find multiple publications that are reporting the associations. And so the the legend here, so the darker color shows the higher number of publications that are reporting associations. And also it highlights to us this genetic variance highlights to us the genes that are implicated in biological pathways that are potentially relevant to the COVID-19 pathogenesis. And that includes the pathways from the viral response, the inflammation, as well as the immune system. So leveraging on the joint collaboration between SG10K Health, Prepare, and Trust, we were able to integrate national COVID-19 case data with our SG10K datasets. So the national COVID-19 records that comprises of information such as the COVID hospitalization and severity, the vaccination, and the test records, and all of these were ingested into the MOH Trust platform. And on the MOH Trust platform, we utilized the HALE Scalable Genomic Analysis Python-based package for all our genomic data analysis and processing. And our colleagues from GIS, they assisted us in the preparation and ingestion of the SG10K dataset onto the platform in the format of HALE matrix tables, which is just a data type that integrates genotype data with its associated metadata, such as the demographics and research phenotypes, the sample and variant QC statistics, and also other SNP annotations. So from the list of data mine variants that we had obtained from surveying the publications, we then followed up with a comparison of the risk allele prevalence as well as allele frequencies in our local data sets. And we did so in an ethnic-specific manner, so we segregated our local data sets into the three main ethnic groups. And so firstly, looking at the prevalence, we see that among the three groups, the Indian group had the highest percentage of risk alleles present, followed by the Malays and then the Chinese. And similarly, for comparisons of allele frequencies, we also observed that the Indian group had the highest correlation with those from the population studies, and this is then followed by the Malays and then the Chinese. And coming to the phenotype data, so for all the SG10K subjects that we were able to align with the national COVID-19 data records, we were then able to derive the severity phenotype according to the level of medical care that is required. So we utilized the 4-category classification here, ranging from uninfected to severe. So uninfected refers to the subjects that are tested negative. And then mild is those subjects that had tested positive, but they do not require hospitalization. And then moderate refers to subjects that were hospitalized. And the severe, if they fulfill any one of the three criteria, such as being admitted to ICU, having requirement for the oxygen therapy, or even succumbing to the disease. So after successfully aligning the phenotype and the genetic data sets, we were then able to do GWAS. So the raw data sets, for the genetic raw data set that was uploaded to the MOH platform, it comprises of 170 million variants and close to 8,000 samples. So we firstly did the data QC steps. So we did the sample QCs and proceeded to segregate the data set into the three main ethnic groups and proceeded to do the variant QCs, whereby we removed the variants that did not satisfy certain criteria. And subsequently, we then followed up with the association analysis using the HALE logistic regression algorithm. So we ran the association for both severity as well as susceptibility to COVID-19. And we classified the phenotypes into binary values to reflect case versus controls. And we also included the covariates in our analysis that includes the vaccination status, the age, the sex, as well as the genetic PCs. We also attempted to apply a multi-ethnic polygenic risk score to our local population and also to verify its accuracy in COVID-19 severity predictions. So the PRS is the risk score that helps us to estimate the genetic risk of an individual of getting severe COVID. And it is calculated by aggregating the effects of many common variants that had been identified from prior association studies. So from our first objective of surveying the publications, we had obtained our list of data mine variants associated with COVID-19 severity. From there, we further selected the variants that had the available information, such as the risk alleles and the odds ratio values, as we would need this information to calculate our polygenic risk score. Then we proceeded to extract the SNPs from our post-QC SG10K genetic datasets that were overlapping with that shortlisted list of variants. And then we could proceed to do our PRS calculation using the standard equation for each of those SG10K subjects. So after calculation of our PRS, we were also able to align those scores with actual disease severity information. And what we found, if looking at the box plots, we can see that the subjects that were having severe COVID, they had higher mean and median PRS compared to subjects that were non-severe. And from the binomial logistic regression that we did for PRS risk categories against the COVID-19 severity, we found that there wasn't a significant association between severity and genetic risk. And we saw that the subjects from the high genetic risk category were having the higher odds of getting severe COVID compared to subjects from the lower genetic risk category. So to summarize, our project was a success, largely due to the collaborative effort that enabled the integration of the COVID phenotypes with our SG10K genetic data set on the TRUST platform. And from the list of data mine variants, we followed up with a comparison of risk allele frequencies, and we found that the Indian group had the highest correlation with those from population studies. We also did a PRS calculation, and we found that COVID severity was significantly associated with the genetic risk. So in future, we hope to expand our analysis to the larger cohorts of 50,000 and the 100,000 cohort, so that we can discover the novel genetic variants to GWAS. And also, we want to extend our analysis to include long COVID outcomes, specifically the cardiovascular and the cerebral vascular complications. So with that, I'd like to end with an acknowledgement of all our project members. Yeah, actually, because our data collection was from year 2020 to 2023, so actually it does comprise of several strains from alpha right up to Omicron. But if we were to try to break up split up the sample set according to variants, then that also would lead to quite a reduction in the sample size. So, so there is a little bit of restriction there. Yeah. Covariate. May not have information on the strain, but it's basically like restricted to the date of collections, like when were they tested positive. Then that we had to infer that, okay, maybe during this date range, this is this variant, and this date range is this variant. Yeah, so during that 3 years of data collection, so most of the samples were actually vaccinated, but because there are many numbers of vaccination ranging from unvaccinated to partially, fully, and then boosted 1, boosted 2, boosted 3. So we sort of use the ordinal scale from 0 to 5 or 6 to quantify the number of vaccinations that that a subject has. And that was one of the covariates that we included into our GWAS and the PRS.
Our next presenter. That was ending slide. Sorry. Thank you.
Hello, good afternoon, everyone. It's my pleasure to be here. It's been an honor to learn from the precise IHCC colleagues. Today, I'm very pleased to share with you the preliminary findings for our investigation on the dietary risk factor for visceral adiposity in multi-ethnic Asian population. My name is Theresia Mina. Today, I'm representing NTU Lee Kong Chian School of Medicine, the Helios SG 100 case study, and precise. As some of you may have already known, visceral adiposity, or colloquially known as tummy fat, is an independent risk factor for mortality. If you have raised visceral adiposity, you're at higher risk of developing type 2 diabetes, hypertension, and hyperlipidemia, as well as cardiovascular diseases. Now, visceral adiposity varies greatly across ancestral group, and in particular, there's also a regional variability in Asia. On the left-hand side is the summary data derived from the UK Biobank for more than 300,000 people, comparing the BMI, waist circumference, and DEXA-based visceral fat mass index, or VFMI for short, of individuals of Indian ancestries, European, and Chinese ancestries. And you could appreciate that, my slides disappear. Okay. You could appreciate that individuals of Indian ancestries have elevated adiposity. And we observe similar patterns in Singapore. So on the right-hand side is the first 10,000 dataset from the Helios study. Here we compare, on green is in the subgroups of Malay ethnicity, blue is Indian ethnicity, and red the Chinese reference group. Again, you could see clearly that individuals of Malay and Indian heritage have greater adiposity on the overall level as well as at the visceral level, and this is very consistent across age. And so now I suppose you could then say, well, okay, then this is all just about genetics, end of story. That's the end of my presentation. But if from what we have learned so far from all the trans-ancestry GWAS, and if there's anything we can learn from the heritability gap, is that genetic variation alone does not fully explain the problem. And so this is why we have to look upstream on the lifestyle risk factors. And here we live in Asia, and we know that our dietary practices varies a lot according to our religion or our culture. And so we need Asian-specific phenotypic data in order to address visceral adiposity public health burden. And we hope to leverage on the molecular information that we have, discovering population-specific disease mechanism by dissecting the dietary-driven metabolites underlying excess visceral fat. A little bit about our cohort. This is the Helios study. We recruit people from all over Singapore. We are based in Lee Kong Chian School of Medicine. This is adult population cohort, different from Gasto, for those of you who are not familiar yet with Helios study, recruiting age 30 and above. Our ethnic proportion is very similar to Singapore national statistics. Chinese 70%, followed by Indian 80%, followed by Malay 13%, and other ethnic groups. Please feel free to visit our study website. Briefly, all of our participants underwent a very comprehensive physiological assessment, collection of biological samples, and we also administered an extensive health and lifestyle questionnaires and dietary questionnaires. For the purpose of this analysis, I'd like to bring your attention to body fat quantification using whole body DEXA scan. With the DEXA scan, you could obtain information about your body fat, your lean mass, as well as bone. I'm particularly interested in visceral adipose tissue, or VAT for short. VAT, or visceral adipose tissue, is estimated by subtracting the total fat in the lumbar 4 region with the subcutaneous fat in the similar region. When we are investigating visceral fat, it is important that we also take into account individual variation in muscle mass or lean mass. Appendicular lean mass refers to the muscle mass on your arms and legs. It is also important to consider individual variability in physical activity levels. Here in Helios, we use International Physical Activity Questionnaire that is further adjusted by 7-day accelerometry data that are collected in a subset of the participants. Of course, we consider general socio-demographic factors such as age, sex, ethnicity, as well as socio-economic factors, smoking, and income. Briefly, for the downstream analysis of this work, we considered individuals that have a complete dietary data with physiologically plausible total energy intake. They also must have complete DEXA assessment. They also must have complete physical activity questionnaires. Not all blood withdrawals during the study were successful. Some blood withdrawals due to a difficult vein did not work very well, so we have a slightly smaller metabolomic sub-data set compared to the full epidemiological data set for this analysis. If I can bring your attention to the descriptive tables on the right, on average, individuals were aged 52 years old, higher proportion in female with a very similar ethnic distribution compared to the overall cohort, 12% individuals with obesity. And if we zoom in on the individuals with VAT more than 75 centiles, these individuals are likely to be older, male, also, of course, likely to have higher BMI, more than 30 kilogram per meter square, but also likelier to come from individuals of ethnic Malay and Indian ancestries, actively smoking, and have less socio-economic household income. We started this investigation at the epidemiological level. Using a locally developed and validated food frequency questionnaires, we derived 69 dietary indices. This includes macronutrients, individual food groups, very established a priori dietary scores that have been established in Western literature. For example, DASH, Alternative Healthy Eating Index, and Alternative Mediterranean Diet. But also important to consider the specific nature of the underlying population. So we also derive some dietary patterns using hierarchical clustering and PC analysis. And here, the dietary risk factors are exposure and visceral fat is an outcome. 21 out of 69 dietary indices were associated significantly with visceral fat after Bonferroni correction. On the right-hand side, we also wanted to compare the performance of these 21 associations with the corresponding anthropometric or fat indices. So if you can focus on the salmon color pink dot of visceral, representing visceral adipose tissue adjusted by BMI, you could appreciate that the strength of the association and the p-values are much stronger, far stronger compared to the conventional anthropometry, such as waist circumference and waist-hip ratio. And it is also in the opposite direction compared to the fat on the other side of the body. So these are fat coming from legs and arms. That is at the univariate levels. 21 indices are quite a lot to work with, so we wanted to narrow this down further. At the multivariable levels, we apply feature selection, in this case, lasso regression. We applied that to food groups as well as dietary indices, and we could identify 10 independent dietary risk factors that are still associated with visceral fat. We wanted to look at the ethnic distribution of these 10 key dietary risk factors, and we found that Malay subgroups had a greater consumption of Western fast food and processed meat, followed by snacks and sweetened beverages. Indian subgroups, on the other hand, have heightened consumption of flavored rice, nuts, seeds, fruits, and highest level of AMET and AHEI scores, actually. And then at this case, you may think, well, that's a bit paradoxical. That doesn't look like what we understood with the highest burden of the cardiometabolic risk factors in Indian and Malay subgroups. And you're quite right, actually. And that's because this, again, is a good illustration of where we cannot just apply a Western-based dietary indices in the non-Western population. This is why it's important to also consider our local dietary pattern. Now, this is where the metabolomics comes. Food frequency questionnaire or food diary or any kind of questionnaire-based tools that ask people how much you eat is very well known to be inaccurate. It's very prone to recall bias, and this is where metabolomics can help us to link and provide insight into the biological mechanism. Using the Metabolon LC-MS-based metabolite panels, we started with over 1,000 metabolites, and then we found 700 were associated significantly with visceral fat adjusted by BMI. And then if we zoom in further, a further 425 were also associated with the 10 dietary risk factors represented earlier. And if we look at the representation of the dietary risk factors, half were associated with AHEI, followed by AMET, NETs and SID, and the PC3. This is a so-called specific diet pattern derived from this analysis. You could appreciate that across this analytical stage, the superfamily metabolite composition did not change materially. Now, what do we do with the metabolites? If we can align these metabolites according to the 4 quadrants of association with the exposure as well as the outcome variables, on the x-axis is the standard beta, so that's the direction of the association with the exposure variables, and on the y-axis is the direction of the association with the outcome variables. Then you could start to see that, in fact, on the top right-hand quadrant, there's very little metabolites there. So what does that mean? It means, from what we could deduce so far, it seems that the insufficiency of protective food group rather than excess consumption of harmful food group could play a larger role in excess visceral adipose tissue. We then perform a pathway enrichment analysis, and we could see some emerging interesting metabolic pathways, such as caffeine metabolism, followed by glycine, serine, threonine, sphingolipid metabolism, and linoleic acid metabolism. The strength and the limitation. This is a multi-ethnic Asian population with well-characterized phenotypes, and as my colleague Dr. Dorino can testify, he's investigated greatly into the details of the reproducibility of self-reported diet and the metabolite measures. But this is not a perfect, and I hope this is not the end of our story. There is limited causality. This is cross-sectional analysis. There are more that we need to do to better understand the biological processes. We need to perform further analysis, such as annotation of metabolic pathway and genomic region, and I'm looking at my colleagues here to help me, or my bioinformatician colleagues. But there are still lessons that we can draw from these observations so far. Insufficient consumption of protective food group, including veggies and fruits, nuts, and seeds, is a risk factor for excess visceral adipose tissue. It appears that in this population, the hallmark of the rather poor dietary pattern is an excess coffee consumption accompanied by Western fast food, sugar, and snack. And so we need to identify barriers towards consuming these healthy food groups and other behavioral risk factors that underpin the consumption of these harmful food groups in this population. With that, I'd like to end my presentation. I thank my fantastic population and global health group here. Special mention to Dr. Nilanjan Sadu and Dr. Doreen Low, our participants, Metabolon, and please feel free to scan our QR code. These are some of the recent meta-archive publications that colleagues have already published so far. Thank you very much for your time. Happy to take questions. Thank you.
Yeah. So when you talk about these protective metabolites, do you know exactly which foods these are linked to?
So as I've shown earlier in the slides here, so we could identify the food groups as well as the dietary indices that are associated with those metabolites. So yes, we could have a look at that, but whether it is causally linked, that we cannot say at the moment, because this is a cross-sectional analysis, if that answers your question. Yes.
Thank you for the nice presentation. I just want to check. You mentioned that because the dietary, like food theory, all those, isn't very accurate to reflect the patient's lifestyle. So you use metabolite as a way to determine what food they have consumed over the weeks where they participate in the study. So do you all did any association between, like, how related are the metabolites measured and the questionnaire they answered, the food they did?
That's my, I'm looking at my expert colleague there, Dr. Doreen Low, that's her work. It's just here. But I mean, yes, so the short answer to that is that yes, you're right, that we need to do that validation as well. We cannot just say that, okay, all of these metabolites are linked to food, because the metabolites are a mix of what is derived from external environment as well as what we metabolize in our body. So there's also intrinsic metabolites, and so that the bulk of the hard work is to tease out which one is associated with the food and which one is. Yes, Prof. Ilio.
Yeah, well, I wonder whether you analyze or stratified by physical activity, whether there is any indication that food intake and physical type of food, amount of food, and level of physical activity may have some joint effect interaction in determining visceral.
Excellent points. Prof, we have adjusted for physical activity, but we haven't looked at the interaction effect. Yeah, so that's the next action item to do.
Just put the variable in the model,
Yep.
but not look whether
Not yet.
in.
No.
That's
Interest.
something to do, isn't it?
Okay. Thank you.
Right, because that's something we find in other studies where in European populations where medium high level of physical activities have a strong, let's say, impact on whether people develop, we can say visceral because we don't have DEXA, but we have a waist hip ratio, so abdominal. I mean, the abdominal obesity has a quite of clear relationship with level of physical activity for similar level of total calorie intake, for example. Yeah, so that's something to be seen.
Good afternoon, everyone. My name is Wang, a health economist from the South Sea Hawk School of Public Health, National University of Singapore. So after 2.5 days of very complex, genetic and scientific stuff, at least very complex to me. So let's try to explore something different. So today I will talk about study about women's preference for taking tests to predict breast cancer risk. So from a discrete choice experiment. So compared with age-based breast cancer screening, race-based screening is potentially to be more effective and cost-effective in detecting breast cancer. And the recent advancement in the genomic medicine, such as polygenic risk score, so makes personalized breast cancer screening more feasible. But in order for such intervention to be successfully implemented at the ground level, so we have to understand whether women are willing to take tests to know their risk at the first place. So there are some existing discrete choice experiment, or in short DCE studies, examining people's preference for taking tests to predict the risk of developing hereditary cancer. And in Singapore, there is one study examining women's preference for taking single nucleotide polymorphism gene testing to get personalized breast cancer screening. But most of these studies focus on the characteristics of the test. So for example, the accuracy of the test, and also how the tests are conducted. For example, whether it's a buccal swab, blood-based, or maybe conducted in polyclinics or hospital. So the factors that are related to implementation part are less examined. So in this study, we want to do a DCE to deepen our understanding about women's preference for taking tests to know their breast cancer risk. And also, we want to understand what factors can explain the preference heterogeneity. So broadly speaking, we explored two groups of factors. The first one are the demographic factors, including age, race, housing type, education, employment status, and the marital status. And we also explored breast cancer-related factors, including personal history of breast disease, family history of breast disease, and whether they know someone who have breast cancer, and their self-perceived risk of developing breast cancer, as well as the concern about breast cancer relatively to other disease. So the first step of this work, we do some literature review and also consulted researchers and the program administrators who have lots of experience in breast cancer screening programs. And from there, we identified 6 attributes to construct the discrete choice experiment. So this table summarizes the attributes we use. So to start, we have the one-time cost for one-time out-of-pocket cost for the test, and we consider the possible ways to reduce late-stage breast cancer at diagnosis. So including more frequent mammography, screening other than mammography, such as ultrasound, and the medication and the surgery to remove breast. And then we consider the annual out-of-pocket cost for checking for breast cancer and whether they have insurance coverage for early-stage breast cancer. The chance, the risk correlation, meaning if a woman is identified as high risk for breast cancer, then the probability of their first-degree relative, including mother, sister, and daughter, are at high risk. And lastly, the risk communication method, so how the information about their risk are conveyed. So here is an example of the questions that we show to the study participants. So for the participants, they have to imagine if they can do a test to understand their risk of developing breast cancer. And then across these two situations, so under which situation they have a stronger preference to do the test. So for the situation, they are constructed using the 6 attributes I presented in the previous table. And then after they choose the situation that they have a stronger preference to do the test, and then they have to indicate if that's the situation in the real world, so will they really do the test? So this is a 2-stage design for the DCE. So for the sample, we target to have a final sample size of 300 women, and we stratify the sample into 3 strata equally based on age. And we conducted the online survey from November 2023 to December 2023. And we did a pilot test with first 30 participants for us to further refine the questionnaire. And as a quality check, so we only used the response from the participants who took 4 to 15 minutes for the survey. And we also included a dummy question in the questionnaire, meaning so for the dummy question, so the one option is usually dominates the other option. So if the participants choose the wrong option for this question, then we will exclude these participants from the analysis. And for the data, we use the latent class analysis. So basically, it can help us identify the subgroup or latent class within a population based on their preference pattern. So preferences are the same within one class, but different across different classes. And we can also use control variables to predict class membership. So just to have a more concrete example to explain what it is. Say for today, I want to do a study, a latent class analysis to understand people's preference for drink in this room. And then I collect data, so after I do the analysis, so I can profile your preference into 2 groups, so coffee lover and tea lover. And further, what I can do is I can try to predict your class membership based on your demographics. Let's say a male age below 30, then after I do some calculation, then I say, okay, so for this person, 80% of chance he is a coffee lover and 20% of chance tea lover. And for female, age between 30 to 50, 30% of chance being a coffee lover, 70% being a tea lover. So basically, that's the kind of results we can get from the latent class analysis. Try to profile the population and understand the factors affecting their class membership. Okay. So here is an overview of our sample. So at the end, so we have 328 women included in the analysis, and roughly equally distributed across the 3 age group. So 76% are Chinese, about 60% lives in HDB 4 to 5 rooms flat, and 60% they have a university degree or above, and 70% are full-time employed, and 60% are in marriage. And in terms of the breast disease-related factors, so 5% they have prior breast disease, and 20% they have a family history of breast cancer, and 43% they know someone with breast cancer. And in terms of the self-perceived chance of breast cancer, 33% think they are below average, 52% think they are average, and 15% consider they are above average. And lastly, so 60% of women express they have a higher concern for breast cancer compared to the other disease. So the results. So after the analysis, we identified 2 groups of preference. So we label them exposed as test supporters and test non-supporters. So firstly, there are some similarities between their preference. So both groups, they do not want to do the test if surgery is used to prevent late-stage breast cancer. And the second, if insurance for early-stage breast cancer is available, then they are more willing to take the test. And last similarity, so communication method doesn't matter. So no matter how we convey information, so it doesn't really affect the decision in taking the test. And the difference is, so first one, obviously, so for the supporters, they have a overall higher willingness to take a test, and they are also less present sensitive in both in terms of the test cost and also cost for the annual screening. And lastly, so for the supporters, risk correlation doesn't matter, but for the non-supporters, it matters, and it makes them less willing to take tests. So meaning if a woman's risk is high, and then given a woman is a high risk, and then her first degree relative is also high risk with more than 50%. So in this case, the woman don't want to take the test. And next, we try to rank the importance of these attributes. So one with a higher ranking, meaning it's more important in affecting women's decision in taking the test. And the orange bar are for the supporters, and the blue bar are for the non-supporters. So overall, the costs are the most important attributes, like similar to many of our other studies, like financial barrier is a concern. And for the non-cost attribute, so the most important one is the cancer prevention method, followed by whether have cancer insurance, early cancer insurance, risk correlation, and the communication method. But for the cancer insurance, we can also say that's a cost-related attribute because it affects the treatment cost. And here, so we try to show, try to give an idea about the potential uptake of the test. So we constructed two scenarios based on the preference of the non-supporters. So scenario one is the scenario where the non-supporters are most likely to take test. And the scenario two is the scenario where the non-supporters are least likely to take the test. So you can see the uptake rate ranges from 2% to 58%. And we also calculate the uptake rate for supporters correspondingly, ranging from 81% to 98%. But to emphasize, for this kind of number, so we have to know its limitation because the women, they don't really have to take the test. So usually we believe such estimates is an overestimation of the actual uptake in practice. So lastly, we explore how different factors are affecting women's preference or the class membership. So in this figure, we show the odds ratio of a woman being a supporter for the test. A value higher than 1, meaning the woman is more likely to be a supporter, and less than 1, meaning the woman is less likely to be a test supporter. So overall, younger women are more likely to be the supporter, and for women who is employed and married are more likely to be a supporter. And for Chinese, they are more likely to be a non-supporter. And a woman who lives in condo and with a higher education level or a proxy for higher socioeconomic status, they are more likely to be a non-supporter. And in terms of breast disease, so for women who had breast disease and also know people with breast cancer or have a higher concern for breast cancer, so they are more likely to be supporter. Whereas for women with a family history of breast cancer, they are more likely to be a non-supporter. So for this one, we think probably for women who already have a family history, they believe they are high risk already. So there's no point for them to further do a test to confirm their risk. So to summarize, from this study, we found the cost, late-stage cancer prevention method, and the insurance for early-stage cancer affects women's willingness to take the test to understand their risk. And our original hypothesis is the high-risk correlation will incentivize women to take tests, but we found opposite results from our study, particularly for the non-supporters. So we hypothesized probably this could be due to a misunderstanding of this risk correlation and also a fear of knowing. So for this one, we have to do some further studies to understand the reason. And lastly, so even though for this work, we found communication method, it doesn't matter in terms of affecting women's decision to take tests. But in practice, it still matters because it will affect how women interpret the results and their decisions on the follow-up actions. Because knowing their risk is not the end. So they have to know how to use the information to help with their disease management. So that's all for my presentation. And so this work I collaborated with Dr. Li Jingmei and Ho Pei Zhu from our Genome Institute of Singapore A*STAR. And also we get lots of useful input from Dr. Michael Hartman's research team in terms of design of study. And the work is supported by A*STAR Social Science Innovation Seed Fund as well as Precise CIP Fund. Thank you. So any question?
Sorry, it's okay.
Okay.
I wonder if you have looked at the number of children that these women have. I imagine if a younger woman, maybe they're still singles, but suppose there's
Yes, so we didn't really control this verb, so we didn't really ask whether women have children. So I think this argument is exactly as what we originally hypothesized, right? So like if the risk correlation is high, the women may care about their children, care about their parents, and their sisters, so they are more likely to take tests. So but we found opposite. So for this one, I think we have to do some follow-up qualitative studies to understand why they think in this way. So maybe they interpret information wrongly, or maybe they have some other concerns. Yes. So originally we also think in the same way. Like we think about, okay, so for people with a higher socioeconomic status, so they like particularly through the channel of health seeking behavior, so they may want to do the test more. But some potential explanation for this is firstly probably because they have either higher socioeconomic status. So if they get breast cancer, so they have less concerns from financial perspective, so they may not want to do the test. And the second, so we are not sure. So probably for people with higher education, like we like to criticize things. So we just don't want to take tests. Before we really confirm, okay, this is something accurate, something useful for us, then we take tests. But again, so this is something we want to further understand. Why we found why for this study we found the people from higher socioeconomic status, they are more likely to be a non-supporter. So if no questions, I will pass back.
All right, last but not least, we have Pen Hong. Let me get the slide up.
Thank you. Thank you for the opportunity to introduce our work. Today, the title of my report is Genetic Model Predicts Lifelong Cardiometabolic Risk in Women. My name is Pan Hong. I'm holding joint position from Institute for Human Development and Potential and the Bioinformatics Institute from A-STAR. What is the next? Okay. So talking about the human genome, it really takes about more than 20 years for us to understand the heterogeneity of human genome. If you remember, back to 2001, when the first human sequence is published in Science and Nature, the title of the report is Initial Sequence and Analysis of the Human Genome. Okay, after 20 years, back to 2022, when the complete sequence is published in Science, the title got changed. It's a complete sequence of a human genome. So by far, we know that there is a huge diversity of a human genome. And we can know from our report that a polygenic risk factor learned from a particular population may not necessarily applicable to the other population. Okay, and we are lucky. We are now in the biobank era. You can learn from this conference. There are so many well-done, big data biobank are established, like Japan Biobank, UK Biobank, all of us from US, and our Singapore Biobank. So for the millions of the participants and the thousands of phenotypes, it enables us to work on the complex of the human traits from the diversity of human genomics. And from this report, I will showcase how we can take the advantage of the cross-biobank for enhance our research and validation. Okay, so here is our work. We are working on the genetic model which can predict lifelong cardiometabolic risk in women. So we started the analysis from the metabolic syndrome, which is a precondition of the cardiometabolic diseases. And we built up a polygenic model called META-S for metabolic syndrome by the machine learning method from the cohort-centric methodology. And we will demonstrate this polygenic model, GMS, have the prediction power for this before disease onset. And this prediction power, the risk prediction power, can persist the whole lifespan for women and cross generation. Okay, talking about metabolic syndrome, this is a very well-known and very well-defined by a lot of research consortium. Yeah, there are 5 important traits related to metabolic syndrome, like glucose, waist circumference, triglyceride, HDL, and blood pressure. So here is the standard cutoff. For individual, if you have 3 or more past this cutoff, past this 5 cutoff, we say this individual has the metabolic syndrome, which is a precondition of cardiometabolic diseases. And talking about the prevalence of the metabolic syndrome, it is really changed across different age group, sex group, and ancestry group, and the disease and healthy group. We're showing here from the large cohort study to showing here, yeah, and the male and the female. We use the dark red and dark blue to show the prevalence of the metabolic syndrome. You see it is changed. When people getting older, the prevalence is higher, and when people get sick, so this is 63 years old cardiovascular diseases patients, then the metabolic syndrome rate is much higher. Okay, so the metabolic syndrome is very, and here is the big picture of our experimental design. We actually studied the polygenic risk based on multi-cohort. So we are focused on the discovery cohort, which is our local cohort, gastro and espresso. These 2 cohorts are birth perspective cohort. Espresso is recruit woman before deliver baby, so from preconception. And the gastro is recruit a woman from pregnancy. So we follow the measures of the woman pregnancy, baby, till baby growing up, measure their biochemistry, metabolic status, mental status, till the eldest kid in gastro is now 14 years old. So these 2 local cohorts, birth perspective cohort, are small. There are thousands of the mother and the baby pairs, but it's very beautiful because we have very intensive and careful phenotype and with longitudinal measures. And we are starting from our local birth cohort. I'm working on the woman from gastro and the espresso. They are the woman, mother-to-be and mothers at their very healthy stage at the year 20 to 40 years old. And we built up a model to validate the model from Singapore. We try to validate our model in UKB, which is the largest biobank. And we extend our validation to ATTRACT. ATTRACT is our local cardiovascular disease cohort with more than 2,000 patients. And Japan Biobank Japan. For the younger kids, we also studied 1,000 gastro baby. So when we arrange our participants, according to their lifespan, you can see that actually our study covers almost the whole lifespan from birth toward the end of life. and we covered across different and Central Asian and European. So talking about the polygenic model, as I said, because our discovery cohort is small cohort, so we didn't do the GWAS analysis. We are using machine learning. So for machine learning, the most important thing is the features. So SNPs are our features. We are using from the public available SNPs from the publication related to these 5 important metabolic traits from the public available data and from the GWAS statistics. We gathered more than 100,000 SNPs and applied to our woman's genotype and doing the LD pruning. At the end, we focus on 30,000 SNP candidates. This is a pool of the SNPs. Each one of them somehow more or less related to these metabolic traits from different publications. Okay, so yeah, I think you, okay, this is miss one slide. Sorry, it's fine. So we are doing machine learning on the training and testing set and built up the GMS model. So as I said, the GMS model is based on the metabolic syndrome. So first, we would like to check the explanation power of our polygenic risk in the explaining of the metabolic traits. UK European, UK Asian, and a track which is a local patient and the gastro child. And we show the male and the female, and you can see it is obviously that our GMS, which is trained on local Asian women, do not have the explanation power in UKBA European. However, it can explain the metabolic traits in UK Asian and our Singaporean and children. And you see the explanation power is better for women than men. And for the particular traits like waist circumference and body mass index, it can reach about 10% in the R square. So it's yeah, it has the prediction power. And related to this metabolic GMS, which is trained on 20 to 40 years women, do they have the power to predict disease in the elderly cohort? The answer is yes. So in UKB Asian, the average age is 55, track is 63, and Biobank Japan is 58. You can see across this list of the metabolic disease like T2D, CAD, hypertension, heart failure, stroke, and myocardial infarction, we have odds ratio at about 1.5 to 2 to cross different cohort. And for UK Asian, the dominant century are Indian. For a track is a Singaporean, it's a combination of Chinese and Indian, and the Biobank Japan is East Asian. Okay, and here is the slides to show the prediction of multimobility, metabolic multimobility, which is defined as the list of the cardiometabolic disease if have 2 or more co-occur for a particular individual. We say he's in a multimobility case. This plot showing with the genetic risk and age only, we can do the prediction with the similar or comparable AUC up to 70% to the metabolic multimobility in Asian women. If your cohort have the missing data, the luxury measures from education, socioeconomic, physical activity, and others, maybe you can consider use the genetic risk and age only to achieve a reasonable good prediction. And here is, I'm sorry, a kind of, here's the measure on the child. So we try to link the association between the genetic risk of metabolic syndrome with the child development trajectory. Our colleagues find 5 different growth trajectory from the BMI longitudinal measures from year 0 to year 6. For these 5 trajectory, there are 2 elevated trajectory, early acceleration and later acceleration. We found genetic risk is strongly associated with these 2 abnormal trajectory. And we have this MRI image, as you mentioned just now, is the fat department MRI. And we see the metabolic risk, high risk people, individual, when we zoom in a particular, we can see the increased abdominal compartment in the child. Yeah, I'm sorry. Yeah, this is the summary strengths and the limitation and complex traits and multi-ancestry cross cohort, Asian population and the limitation. Yes, the candidate sleeps is mostly from Westerner study, right? And we are also limited by the limited training samples. I have a take-home message. Okay. There exists the genetic signature for metabolic traits that persist for women throughout their lifespan and in their offspring. And here, last but not least, I would like to thank the big project team. Actually, we are work for almost 500,000 of the participants as the data set. I would like to thank all my colleagues and the stake owner of the cohorts and the grant organization, and some of my team members are also sitting here to support me. Thank you very much. Okay, the sleep selection is at least one. Yeah, at least one, so no need to co-occur in different traits. So we accumulate the sleep either in one of these 5 metabolic traits. Thank you. Yeah. Yes, because the discovery cohort is gastro and espresso women. So the machine learning is learned on women's metabolic syndrome. So it is within expectation that we have better performance in women than in men. Thank you very much for the question. So currently, I don't have metabolic syndrome data for men. So for this study, we need the genotype plus metabolic syndrome data. I'm thinking if we can assess precise and some other local data to enhance this model. Absolutely, yeah. Thank you very much.